All right, wrapping up chapter 9, looking at some more about the physiology. Right, now we're looking at the whole muscle. So instead of the individual sarcomeres and how they work, now how does the whole muscle work together? So we call this muscle twitch, which is the response of the muscle fibers by a single action potential coming down that motor neuron that we covered last time. So you have three phases to this twitch. There's the lag or kind of latent stage, and this is from the stimulus to the beginning of the contraction. So it's not an immediate phase. So we see it here. So this lag phase that occurs. So we have the stimulus applied, but it takes a little bit before that contraction, so called the lag phase. Then we have the contraction, that's that release of calcium, where those cross bridges are going to form. And then we have the relaxation, where the calcium returns to the sarcoplasmic reticulum. All right, we have two different types of muscle contractions, isometric and isotonic. And the difference here is about the length. So it's isometric, so here thinking about metric is the length, so there's no change in length, but the tension is increasing on the muscle. So this would be like our postural muscles. So the length, they're not shortening or lengthening, but they are becoming more tense. Whereas isotonic, these are actually changing the length, but the tension remains constant. So again, with both of them, something's saying the same. It's just whether it's the tension or the length. All right, now looking at motor units, and a motor unit is defined as a single motor neuron and all the muscle fibers that it's attached to. So again, our motor neurons coming down, the axon terminals kind of branch off so that we have different neuromuscular junctions here. So each of these muscle fibers, again, made up of those little individual myofibrils, each of them has a different axon terminal attached to them. But again, one motor neuron can come down and branch so that it's attached to multiple muscle fibers. So all the muscle fibers in that one motor neuron make up the motor unit. So an action potential in the neuron is going to cause all of those muscle fibers to contract. And if we look at just some numbers to go along with it, large muscles have motor neurons with many different muscle fibers. Smaller muscles that have like delicate movements, those are going to be attached, where we have one motor neuron just attached to a few muscle fibers. So the bigger the motor or the muscle is, the more muscle fibers are attached to a single neuron. Whereas the smaller ones that we need some really delicate, intricate movements, we have fewer muscle fibers connected to one motor unit so that they can be controlled more individually. Now looking at the force of the contraction, so we know how the contraction occurs with the calcium coming in and the sliding filaments, but the strength of that contraction varies anywhere from a weak contraction to a strong contraction. So we have this graded fashion, so this intermediate in between. So it's not just all or nothing. We have weak or strong contractions, and it depends on that force that's generated in those individual muscle fibers. So by increasing the number of cross bridges where the actin and myosin are binding when calcium is present, that allows the fibers to have more force. So if you remember the picture, all along the actin, there are all of those active sites where the myosin heads can attach. Okay, well, if just a few of them attach, then we have a weaker force. If we have a lot of myosin heads attaching, then we have a stronger force. So by increasing the number of cross bridges, we can also increase the frequency, the diameter of the muscle fiber, and then also the length, or the muscle fiber length, can also increase it. So all three of these things can increase the number of cross bridges. Again, how much they're stimulated. So again, the motor neuron constantly sending out acetylcholine, the thickness of the muscle fiber, and the length of time of the contraction, so how long it's contracting, all of those increase the cross bridges. All right, so if we look first at the frequency of stimulation as a way of increasing those cross bridges. So this is referred to as TREPA, which is a French term, pardon my pronunciation there. So essentially this is due to the calcium that's in it. So once we have one response, so the calcium increases from that first one, well, it's not completely removed into the sarcoplasma before that second stimulus can occur. Therefore, there's already some calcium in there. So during the next contraction, the calcium concentration increases slightly, And it makes the concentration more efficient because we have increased cross bridges, because there was already some calcium present to begin with. And this is the whole idea behind warming up before different athletic events or before exercising, is because the warm up increases the frequency of stimulation, which increases, again, we have more calcium available, so we have more cross bridges, and it makes those contractions stronger. It's called a wave of summation in tetanus. So tetanus, we think about like our tetanus shot. Well, it's because of this muscle contraction that we get. So as the frequency of those action potentials increases beyond that trepe, then the contractions, again, occur with greater force. So we have all of that kind of calcium buildup in there, staying there, in more cross bridges. So this wave of summation, this is the muscle tension increases as that frequency increases, again, due to those more cross bridges. So we can also have incomplete or complete tetanus. So incomplete is where the muscle fibers partially relax between contraction, and complete tetanus is where there's no relaxation. So because the frequency is coming, because it's so frequent, there's not a time for relaxation. That calcium is not being removed, so they essentially stay contracted. Now, this tetanus is what led to the naming of the disease that causes the disease tetanus, is because those muscles stay contracted. They're not relaxing due to an infection. So again, tetanus is a continuous contraction of the muscle. So this wave of summation and tetanus result because of mechanical events of the muscle twitches. Again, they take longer than the electrical events. So again, the actual moving of the myosin heads off of there and everything, that is going to be a much slower effect than what all the electrical signals coming in. So it allows several action potentials to be generated in the time it takes for a single twitch, so a single contraction of that muscle. And again, that's because the calcium ions cannot be removed fast enough, so the muscle fiber is unable to relax. And this is just kind of showing what's happening during this. So again, our single action potential arrives at the fibers. Again, it causes the contraction or the twitch that completely relax before the next wave. So in this scenario where frequency, we're getting contraction, and then it comes all the way back down to rest. So it's going up and then down. So we're getting complete relaxation in this twitch. However, with wave summation, whenever our frequency is coming in, here you see there's not complete relaxation before we get another stimulus. And then another stimulus. So it's never coming back down to that relaxed state. Then if we get more and more frequency, a higher frequency here, now we get that incomplete tetanus to where it's coming up. It's relaxing a little bit, but nothing close to completely relax. And then finally, the complete tetanus, it's just staying contraction. It's not even going down or releasing at all. All right, now what about the muscle fiber diameter? How does that impact our contraction? So the greater the diameter, so the thicker, the wider that it is, the greater the force. So again, wider, thicker muscle fibers are going to generate more force than what the smaller ones. So in general, larger diameter fibers have more myofibrils, and they can form more cross bridges. So the more there are, again, more cross bridges, the more the contraction. Now the muscle length at the time of contraction, so we have active tension, we also have passive tension. So with active tension, the force applied to the object is the force applied to the object to be lifted when the muscle contracts. So this active tension increases or decreases as the muscle fiber length changes. So this is referred to as the active tension curve. So if it gets stretched too far, then there are fewer cross bridges. So if they go too far past each other. If it's not stretched at all, again, those thick filaments touch the Z disc, and there's very little contraction. So think about that sliding, the sliding filaments. Again, if we go too far, we decrease contractions. Not far enough, we don't have enough of those cross bridges. Now with passive tension, this is the tension applied to load when the muscle stretches but is not stimulated. So our muscle is stretched, but it's not being stimulated by those neurons. So we refer to that as passive. And then total tension is whenever we add both of those together. And here we can just visualize the effect of that muscle length on the tension. So in this scenario, in the muscle length 1, the actin and myosin are already slid across each other, so there's not a lot of distance. They can't shorten much further, so that contraction is not going to be as much because there's not as far to go. Muscle 2 length here, this is the prime, so this is going to be the optimal sarcomere length. This is where they're spaced far enough apart that we have that H zone to get started, so we can bring them closer together. We have a lot of sliding, and we get that optimal contraction. Muscle length 3 is the opposite of 1. Here they're too far apart, so they're stretched too much, so we're not going to be able to generate enough sliding here to increase those cross bridges. All right, so now what about that force of contraction again in the whole muscle? So the strength of the contraction again is graded, so it ranges as we mentioned. And then we can also get this multiple motor unit recruitment. So the strength of the contraction depends on the recruitment of motor units. So again, within a muscle, there's more than one motor unit. Again, motor unit is the motor neuron coming down and the muscle fibers it's attached to. So that's one motor unit. So if we just have one working together, we get some contraction. But if we have 5 of them working together, we get a much stronger contraction. So the more motor units we have that get activated, the greater the force of that contraction is going to be. So again, here's our threshold for stimulus. So this is one motor unit that gets activated. We get some tension, we get some contraction. But as we increase the number of motor units getting activated, our tension, our force of contraction is increasing much greater. Now it does reach a point where it plateaus, where it can't contract anymore, even if more and more motor units are recruited. So how does this recruitment work? So there's a subthreshold stimulus. So at this point, there's no action potential, which means there's no contraction. Our threshold, again, like we talked about with the action potential, that's when it actually gets generated, which leads to the calcium coming in and we get contraction. Submaximal stimuli, those are stronger stimuli that produce action potentials and axons of additional motor units. And then maximal stimuli, these are action potentials produced in axons of all motor units of a muscle. So again, this will create the greatest stimulus possible. Even a super maximal stimulus at this point has no effect. So at the maximal, again, that's what I was talking about, the threshold. It does reach a point that even if you engage more and more motor units, they can't contract anymore. So submaximal is somewhere in between the threshold, where we get contraction, and the maximal. So as we recruit more and more motor units, we're getting a greater contraction. So the size principle, so this is during the recruitment. So as motor units are being incorporated, small motor units are recruited first, and then the larger ones. And again, small just means one motor neuron to a couple myofibrils, whereas large would have multiple myofibrils attached. And then muscle tone is that constant tension by muscles for long periods of time. So this is due to small percentages of motor units contracting out of phase with one another. So they're not being recruited properly. So they're, again, one unit and then another, but not at the same time. Now, if we look at these isotonic contractions, again, isotonic, we're changing the length, keeping tension the same, so same tension. So we have concentric and eccentric isotonic contractions. Concentric, this overcomes that opposing resistance and the muscle shortens. Eccentric, the tension is maintained, but the opposing resistance is great enough that it causes the muscle to lengthen, right? So again, with concentric, the muscle is shortening. With eccentric, the muscle is lengthening. So back to our twitches here. So these types, so we have a slow twitch, which is type 1, and a fast twitch, which is 2. So slow, as it implies, it contracts more slowly. These are going to have smaller diameter, so less cross bridges. They do have better blood supply. They have more mitochondria to provide that ATP They are more fatigue resistant than the fast twitch. So this is more like slow and steady wins the race kind of thing. So it's more of a constant, steady tension that can be applied. And they have a large amount of that myoglobin. So these help with more of our posture, more in lower than the upper limbs. This is also just to kind of put it in a darker tissue due to having more of that myoglobin produces a darker color. So this is our dark meat. So again, this is more kind of that tension, the toning that occurs, more sustained. So endurance runners, they would want to develop their slow twitch muscle fibers more so than fast twitch. And those fast twitch, again, as the name implies, they're rapidly responding to stimuli. They have the myosin that can break down the ATP more quickly, so they can, again, continuously contract much faster. They have less blood supply, though, and so they have fewer and smaller mitochondria. So that means that they have less oxygen coming in, less mitochondria to make ATP, so they're going to fatigue much faster. So because of all the extra mitochondria and the extra blood supply and oxygen coming into slow twitch, again, they have a continuous supply of energy. Fast twitch are faster, but they can't maintain that very long. So this will be the lower limbs of our sprinters, upper limbs of most people, and again, our white meat. And this comes from that oxidative and anaerobic form. So that anaerobic condition where we switch to fermentation and produce the lactate, and that builds up, and that's what makes us sore, is due to these fast twitch muscle fibers. So a little bit more about these. So most of the muscles have both types, but they vary from person to person. Conversion from one type to another is not easily done. So typically once a muscle fiber is slow twitch, it stays that way. Fast twitch stays that way. Now, what effect does exercise have on that? So it changes the size of the muscle fiber. So again, we don't really change the number of muscle fibers after development. Again, it's more the size of them. That's what exercise is doing. So hypertrophy, this increases the muscle size, which is increasing the myofibrils, increases the nuclei, so we have more fusion that's happening, and it increases in strength because we have better coordination of the muscles. Enzymes are working better, we have better circulation that's occurring. Then atrophy, the opposite of that would be the decrease in muscle size. So this is the reverse. Again, in severe situations, the cells can actually die in this case. So this would be a loss of muscle tone, a loss of muscle contraction. Now, heat is another one of the functions for muscles, one we don't really think about other than like shivering. So during exercise, that metabolic rate and heat production increase. So not all energy is converted. It's not efficient. So some gets lost as heat. Even after exercise, the metabolic rate stays high because it's trying to replace the oxygen. Due to the oxygen debt, it's trying to replace the glycogen that was lost that stores all of our energy for us. And that excess heat lost because of the vasodilation and sweating as we're trying to regulate our temperature. And then again, shivering is that uncoordinated contraction. But again, that shaking is altering those muscles, causing contraction for the purpose of generating heat. All right, now what about the energy sources? So I kind of mentioned a little bit about this already, but essentially there are three major ATP-dependent enzymes. We have one on the myosin head, so it can form the cross bridge. They have the sodium-potassium pump that we mentioned that restores the resting membrane potential, and that calcium reuptake in the sarcoplasmic reticulum. Again, we've mentioned these already as far as why we need ATP. So muscle fibers only have about enough ATP to last for about 5 to 6 seconds. So the need to take in more is constant. So where does this energy come from? So the conversion of 2 ADP to 1 ATP, so we can kind of combine two together to 2 ADPs to form 1 ATP and then an AMP. So just a quick refresher, adenosine triphosphate has 3 phosphates on it. It has the most energy. ADP has 2 phosphates, a little less energy than ATP. and then AMP only has one phosphate, which has the least energy. So two of those can be, two ADPs can be combined to make one 3-phosphate molecule and one 1-phosphate. That uses an enzyme called adenylate kinase. And we can also transfer phosphate from an enzyme called creatine kinase. That's added to ADP to form ATP using an enzyme called, again, creatine kinase. This is the idea of using, a lot of muscle builders take in creatine, and this is why. So they have that creatine there to transfer the phosphate to ADP. Anaerobic respiration, so this occurs in the absence of oxygen. So now cells are breaking down glucose, but because oxygen isn't present because they've run out, now cells switch to fermentation, which produces that lactic acid. That's what causes soreness to occur. And really the term anaerobic respiration is not very accurate. It is unfortunately what your book uses, but this really should be fermentation is really what this process is, because there's no electron transport to give it the name respiration. It is anaerobic, but it's really should be referred to as fermentation. And then we have aerobic respiration. This requires oxygen. It fully breaks glucose down into carbon dioxide and water and produces huge amounts of ATP. So again, it's much more efficient. But again, as cells run out of oxygen, they can keep up with some demand using fermentation. All right, and this is showing that ATP. So again, with adenylate kinase, we're taking 2 ADP molecules here, which have 4 total phosphates. Now we're making 1 ATP with 3, and then 1 AMP. Our creatine kinase is just taking a phosphate off creatine and adding it on to ADP. Whenever it gets added, we make ATP. Again, anaerobic glucose goes through glycolysis, produces pyruvate, which generates energy, but then we need to do something with that pyruvate. So that fermentation process without oxygen is going to allow glycolysis to continue, but with very little ATP. The benefit, though, is that no oxygen is needed. And then in the aerobic, we do glycolysis, pyruvate to get our pyruvate. Then pyruvate goes on to the mitochondria through additional steps that require oxygen, completely breaks the glucose down to carbon dioxide, and we get huge amounts of ATP. All right, so what happens whenever our muscles fatigue? So this is defined as the decreased capacity to work or reduced efficiency. So the mechanisms here, so we can get acidosis. So again, as that lactic acid builds up, that acid is going to alter our proteins and their ability to function, running out of ATP. So these are either due to an increase in consumption or a decrease in production. Oxidative stress can occur. This is characterized by reactive oxygen species. This occurs as mitochondria are going through aerobic respiration. It releases some of these free radicals that can ultimately be damaging. So again, as it continues to work and work and work, we build up those oxidative molecules. And then also local inflammation. So physiological contracture. This is a state of fatigue that's due to a lack of ATP. So neither contraction nor relaxation can occur. Then psychological fatigue is actually the most common type, and this comes from our central nervous system rather than actually from our muscles. So again, more psychological, more due to the nervous system's ability to contract muscles. Physiological means they cannot contract or relax. Now, rigor mortis and muscle soreness. So that rigor mortis is after death. So this is whenever the muscles become very rigid. And this is due, and it does take a little bit of time, and that's because there's still some buildup, some storage of calcium, but as calcium leaks into the sarcoplasm, again, it attaches and those cross bridges are going to form, but the rigor ends as the tissue deteriorates. But essentially what happens is the calcium's still there, it starts leaking out after death, so it's just like it's contracting, so the calcium causes the cross bridges to form, but now there's no ATP to release the myosin so it stays contracted. And it stays that way essentially until the tissue just deteriorates and the proteins break down and the cross bridges end. The soreness I mentioned a little bit already, but again, this is due to those inflammatory chemicals that can occur, but also due to that lactic acid that can build up as well. Now, what happens whenever we have oxygen deficiency or excessive post-exercise consumption of oxygen? So first of all, just deficiency means, again, that lag time between when a person begins and when he or she begins to breathe more heavily. So again, as we start exercising and, and we're just kind of walking, we can keep up with oxygen. But if we start running, now our ability to keep up with oxygen decreases. So we start breathing more heavily, trying to bring more and more oxygen in. Now, excess post-exercise consumption, so that this is the lag time before breathing returns to the normal once you stop. So whenever somebody starts exercising, they start running, it doesn't take long, they start breathing heavily. As soon as they stop running, they don't automatically just stop breathing heavily. So that's the post-exercise oxygen consumption. So that deficit has to be replayed so that metabolic rate stays high to restore homeostasis. All right, now just looking at the other couple tissues. So we spent a lot of time talking about skeletal muscles, but just a little bit here to wrap up with our smooth muscles. Again, not striated. These fibers are much smaller than the skeletal muscles. They have that kind of spindle shape with a single nucleus. They do have more actin than myosin, myofibrils, microfilaments. They have caveoli, which are indentations in the sarcolemma, again, that plasma membrane of the muscle. They act kind of like those T-tubules that we'd seen with the skeletal. They have what are called dense bodies instead of the Z-disc that we saw in the skeletal muscle. So the actin attaches to these dense bodies instead of to the Z-disc. So they have non-contractile intermediate filaments in addition to actin and myosin. And the calcium required to initiate contraction, it binds to, instead of that like tropomyosin and troponin, instead of binding to that, it binds to a molecule called calmodulin, and that regulates myosin kinase to form those cross bridges. So again, calcium is still what's triggering it. The calcium just works a little differently. And then relaxation occurs because of an enzyme that removes the phosphate from the myosin. Right, and again, just kind of looking at this, so again, single nuclei that's here. We have the spindle shape to where it's kind of tapered there at the end. They're packed on top of each other though, and again, not striated. All right, so a couple different types of smooth muscle, visceral and multi-unit. Visceral are in sheets and they act as a single unit. So a lot of different gap junctions that allows that communication and kind of a wave of contraction. Once one part undergoes contraction, those gap junctions allow the calcium to diffuse and contract in waves. So these are typically autorhythmic, and the synapses are a series of dilations along those branching axons. Now the multi-unit smooth muscle, these are cells or groups act as independent units. So working together, visceral, working independently, multi-unit. So these are more in sheets with blood vessels. They have bundles, which are our recti pili that make our hair stand on end. We also have single cells like the capsules of our spleen. So these have fewer gap junctions, and the synapses are more similar to the skeletal muscle. All right, and then finally our cardiac muscle, again, only in the heart. We do have striations like skeletal, but it's more branched. Typically only one nucleus, so they're not joining together into like multiple cells fusing into one. They have what are called intercalated disc and gap junctions for that communication, again, autorhythmic. The action potentials of longer duration, they also have a longer refractory period where they're going back to normal. So both sodium and calcium influx are needed for that depolarization. So it's not just sodium coming in, calcium also has to come in to cause depolarization, so the membrane potential becoming less negative, more positive. And then the calcium regulates the contraction, which is what we'd seen with the others. Again, this is what these guys look like. So again, single nucleus striations, but we get more of this branched structure that's going on here. All right, and that wraps up our muscle discussion.